The cell cycle is a cycle that cells undergo in order to duplicate. By duplicating its cells, the body may grow. The duplication of the cells is also useful for repair of damaged tissues and asexual reproduction. The cell cycle consists of three stages known as interphase, mitosis, and cytokinesis. Interphase is divided into three parts known as G1, S, and G2. G1 is known as the period of growth where the cell grows to its full size. During S, the DNA of the nucleus in the cell is replicated. In G2, the cell prepares itself to begin mitosis. Mitosis is the process of nuclear division. There are four distinct phases during mitosis, those being prophase, metaphase, anaphase, and telophase. During prophase, the duplicated chromatid network becomes chromosomes. The chromosomes consist of two identical strands, the chromatids joined by a centromere. The nuclear membrane and the nucleolus disappear and the centrioles move to opposite poles of the cell and begin to develop spindle fibers. During metaphase, the chromosomes move to the equator and arrange themselves in a single row on the equator of the cell. The spindle fibers then attach to the centromere of the chromosomes. During anaphase, the centromere of each chromosome divides into two. Chromatids separate and move to opposite poles of the cell. The chromatids are now known as daughter chromosomes and are both exactly identical. Telophase is the final phase of mitosis. The daughter chromosomes arrange themselves at opposite poles of the cell and the spindle fibers disappear. Two separate nuclear membranes form around each group of daughter chromosomes and a nucleolus is formed within each nucleus. Cytokinesis is the last stage of cell division. During this stage, the cytoplasm divides and the cell separates from one another, forming two new cells. Cell division is regulated by cell cycle clocks. The cell cycle is regulated so that when a cell needs to divide, it is able to integrate signals that the cell receives from its neighbors to determine whether or not it needs to duplicate. If it should duplicate, then it begins the cell cycle, and if not, it remains temporarily or permanently unduplicated. Proto-oncogenes that encourage cell division and tumor suppressor genes that prevent cell division are two types of genes that play a large role in regulating the cell's duplication. In order to maintain a balance, there must be an equal amount of proto-oncogenes and tumor suppressor genes in each cell. When there is an imbalance, for example, if there are more proto-oncogenes than there are tumor suppressor genes, then it becomes possible for cancer to form. This imbalance takes place because of mutations that create a change in the cell's DNA, and therefore disrupt the balance of the genes, ruining the regulation system of the cell and causing it to divide uncontrollably.